Hello students, welcome to the class. So before going to start the class, can you tell me whether I'm audible and the screen is visible? Yeah, very good. Okay, so let us start the class, okay? Okay. Okay. So before going to start the class, here is a question for you all. Okay. So can you try to answer this, guys? The question is, the biomolecules are, okay, option A, they are telling that. So biomolecules are inorganic material only. Option B, organic material only. Option C, all the carbon compounds obtained from living tissue. Okay, then option D is only DNA and RNA. Student, whenever you are answering the question, first eliminate the wrong options. Okay. Okay, let us try. I will run the poll. You can answer me in the poll. Okay. Yeah. You can answer me in the poll or you can answer me in the chat box also. Yeah, you can answer me in the chat box. No problem. I will end the poll. Answer me in the chat box. Answer me in the chat box. Yeah. So students, whenever we are talking about the biomolecules, so we know that during the evolution, what happened? So first, the smaller molecule will form, then the organic molecule forms, right? So yes. So the correct option is option C. Okay, our body is made of all the biomolecules, right? For example, if you take a carbohydrate, protein, lipids, nucleic acid, so these all are the biomolecules, right? If you take a body tissue or if you take any plant tissue, you can see the biomolecules in that. Okay. So we will eliminate the wrong option. Inorganic material only. No. So student, here you have to remember whenever there is only is given, so you have to first check for there is a clue that is okay. So it may be a correct or not maybe. Okay. So only I will eliminate this also. No, not only. And only DNA and RNA. No, DNA and RNA is a genetic material, right? Other than there is a biomolecules, are there protein and carbohydrate, lipids, all the things. So that's why option D is not correct. Option B is not correct. Option B is not correct. A is also not correct. So C is the correct answer. All the carbon compound obtained from the living tissue. Either it may be a plant or either it may be a animals. Okay. Okay. Then here is another question for you based on the enzymes. Okay. Yeah. So which enzyme was first isolated and purified in the crystal form, in the form of crystal? Okay. In the form of crystal. Can you answer me, guys, in the chat box? I need answer from you. Okay. So you must be interactive in the class. Okay. Shagufa Mushraf, can you tell me which is the correct answer here? Can you try to answer? Yeah, try to answer. It is a very easy question. Can you answer me, student, for this question?
Ma'am, urease.
1 minute. Very good. Yes. So urease, right? Yes. Urease is the correct answer. Urease, right? Yes. Not amylase, not ribonuclease, and pepsin also. So amylase, you know, right? Where it is used, the amylase. Amylase in the saliva, it will break down the carbohydrate. Ribonuclease, where the ribonuclease will work, student? It will cleave the RNAs, right? Ribonuclease. Pepsin, it will cleave the protein. Okay. So then we will talk about what is this biomolecule. Okay. So our body is made up of the biomolecule, right? So biomolecules are chemical compounds found in the living organism, right? So in the living organism, the organic chemical molecules will be found carbon containing. So those we will call it as a biomolecules. Okay, all the biomolecules is consisting of the carbon. Okay, yeah, they include the organic and inorganic compound also, right? When we are talking about the nucleic acid, in the nucleic acid, the phosphate group is also there, carbon group is also there, right? Yes. So when we're talking about the protein, in the protein we can see the ammonia, right? Yes. So in the protein we can see the nitrogen group, right? So this all the thing, carbon group is also there, whereas the inorganic compound, so that is a phosphate and nitrogen compounds are also there. So that's why it is consisting of both organic and inorganic compounds. Okay. Okay, let us talk about metabolites first. Okay, so what is this metabolites, students? Actually, metabolites. First, we will talk about what is metabolism. Okay, so first we will talk about what is the metabolism. Okay, so have you know, student, there are the two types of the metabolisms are there. One is anabolism, right? One is anabolism and another one is catabolism, right? Catabolism. So this anabolism and catabolism will happen in body itself, right? Whenever this anabolism is there, means the synthesis of the biomolecules will take place. Synthesis. Anabolism is nothing but synthesis. Okay. Synthesis, or you can call it as the biosynthesis, or yeah, biosynthesis. Biosynthesis. For example, you know that every day or every minute the protein was synthesized in our body, right? So the protein is synthesized from the smaller molecule, that is the amino acids, right? So that is what we will call it as a anabolism. Okay, anabolism is a biosynthesis process. Okay, then we'll come to the catabolism. What do you mean by catabolism, student? Catabolism means destruction. For example, if you remember, if you just think that one building is there, building is demolishing, right? So that thing we'll call it as a catabolism. For example, you can take an example of carbohydrate. Okay, carbohydrate. The breakdown of the carbohydrate is called as, in which process the breakdown of the carbohydrate will take place, student? Can you tell me that process name? Can you tell me that process name? Do you remember glycolysis? Do you remember glycolysis? What will happen in the glycolysis? Do you remember what happened in the glycolysis, student? In the glycolysis, the catabolism is nothing but breakdown. Okay, breakdown. Okay, in the catabolism, you see here in the glycolysis, what will happen? Glycolysis. Glycolysis, only telling it is glucose is breakdown into the pyruvate, right? Glucose is breakdown into the pyruvate. Then you are getting the ATP, energy molecule, right? ATP plus pyruvate you are getting, right? So that will call it as a constructive, that is called it as a destructive. Okay, so remember like this. Okay, CD, catabolism means destructive, anabolism means biosynthesis. Okay, anabolism means biosynthesis, catabolism means destructive. Okay, student? Okay, so here you can take an example of protein synthesis. Protein synthesis. Okay, in our body, the protein is synthesized, right? So that is called as a protein synthesis. Okay. So then coming to the primary metabolites and the secondary metabolites. Okay. So let us talk about in the body, there will be a primary metabolites and secondary metabolites. Can you tell me what is the primary metabolite? This primary metabolites is going to be synthesized only in the plants, fungi, and some bacteria. Okay. So this is the primary metabolites. 1 minute. Yeah. See here. Primary metabolites. Primary metabolites. It is going to be synthesized in all the organisms. Not in plants and only bacteria and fungi. It is going to be synthesized in all the organisms. Okay, student. Primary metabolites is required for the growth. Can you think, student? You have learned in the first PU. Can you recall what are the things you have studied? Reproduction, growth, reproduction, and survival of the organism. Okay. for the survival. This first primary metabolites is very, very important for growth, reproduction, and the survival. Can you think which are the primary metabolites? Can you think which are the primary metabolites, student? Yes, yes. You know the answer, right? Proteins, right? Without the protein, no growth is going to be happen. Without the protein, no reproduction, right? Protein and carbohydrate, protein, carbohydrate, and nucleic acid, and lipids. Okay, so these are the things we will call it as a primary metabolites. Okay, so then we'll coming to the secondary metabolites. What do you mean by the secondary metabolites? Secondary metabolites, we are kept in the second stage. That means it does not require for growth, reproduction, and survival. Okay, it is not required for, it is not at all required for growth. Okay, then reproduction and also for the survival of the organism. Okay, but it is required for defense mechanism. Okay, so why it is required? It is required for defense mechanism. Okay, for the defensive purpose, the secondary metabolites is required. For example, if you take student, here the plant from the plant, bacteria and fungi, it will release as the secondary metabolite. Whereas without this secondary metabolite, the plant, fungi and bacteria will grow, it will survive, it will reproduce. But it is required for only for the defense mechanism. Defense means what? To survive from the other organism. Okay, that is called as the defense mechanism. Okay, secondary metabolites. So then here there are some examples are given for the secondary metabolism. This is the important thing. In the exam, they may ask you in the multiple choice. Okay, so just remember pigments. These are the secondary metabolites. Okay, when we come to the pigments like carotenoids and anthocyanin, and when it comes to the alkaloids, morphine and codeine. Okay, so morphine, codeine, and caffeine. So these all the things are secondary metabolites, and it will comes under the alkaloids. So student, here you have to remember alkaloids will end in INEN. Okay, so see here INE. Okay, here also see codeine INE. Okay, you can remember like this. Okay, morphine and the codeine. Okay, so then these are the narcotics drug. Okay, this is the narcotics. Okay, let us come to the terpenoids. So what are these terpenoids? So these terpenoids are usually fatty acid things. Okay, fatty acid containing things. So it will be modified into the monoterpenes and the diterpenes. Okay, and coming to the essential oils. So you know, in the lemon, whenever we touch the lemon, some types of the odor will come, right? So that is the because of the oil. Okay, lemon smell. Okay, lemon smell is due to the lemon grass oil. Okay, lemongrass oil. And you can see the mint. From the mint also you'll get the smell, right? So those all the secondary metabolites. Okay, mint also. And you can take the toxin. So toxins which kill the other organisms, right? So you can take the toxins, abrine and the ricins. Okay, abrine and the ricin. And lectins you can take. Lectin means it is a protein. It will come and bind to the carbohydrate. Okay, so lectin will come and bind to the carbohydrate. So it is usually used in the purification of the carbohydrate. 1 minute. Okay, so here lectin, so that is the concovalin they are using. Concovalin, they will go and bind to the carbohydrate. It is used in the purification of carbohydrate or separation. For example, if you take some solution you have, in that solution you have to separate the carbohydrate. Okay, on that time you can use a concovalin. Concovalin will go and bind to the carbohydrate. Then you separate the concovalin. Along with the concovalin, you separate the carbohydrate also. Okay, like that it is going to be work. Okay, so then coming to the drugs. Okay, so there is a vinblastine and curcumin, and also the vincristine is the drugs. Okay, these drugs are useful in cancer treatment. Okay. Have you learned, have you studied the cancer chapter? Have you studied the cancer chapter, students? In that, it will come, okay? Vinblastine and the vincristine and curcumin are used in the cancer treatment. Cancer treatment, vinblastine, okay? And another one is there, so that is called as a vincristine. Vincristine, okay? Cancer treatment. It is used in the cancer treatment. And then we will go to the polymeric substances. Polymeric substances are rubber, gums, and the cellulose. So these all the things are secondary metabolites, okay? Secondary metabolites of the plants, okay? So this is about the secondary, this is about the metabolites. In this table is very important. You have to remember this, okay? In the multiple choice, they may ask you. Do you have any confusion here, student? If you have any confusion, you can ask me in the chat box, or you can tell me, yes, student, be active in the class, okay? So whenever we are asking the question, please answer me in the class. Otherwise, how can I think that you are listening to the class? No? Answer to my questions. Then I came to know that at least you are listening to the class. No. That too, we are talking about the very interesting chapter, that is biomolecules. Right? Okay. So then we will move to the biomolecules. So student, whenever we are talking about the biomolecules, so there are two types, okay? One is macromolecule and another one is the micromolecule, okay? When we are talking about the biomolecules, here there are two types, okay? One is the macromolecule. Macro means what? Having the big size, right? Macromolecules, and another one is the micromolecules. Another one is the micromolecule, okay? Whenever we are talking about the macromolecules, so these molecules having the molecular weight greater than the 1000 Daltons, okay? Greater than the 1000 Daltons, okay? These are having the molecular weight, greater molecular weight, so that is the 1000 Daltons, okay? So whenever we are talking about the micromolecules, so this is having the molecular weight that is below 1000 Dalton, okay? Below 1000 Dalton, okay? So now I need an answer from you all, okay? Can you tell me which are the examples for macromolecules? Can you tell me, student, which are the examples can we give for the macromolecules? Okay, so let us think. Big molecules I'm telling. In the biomolecules, which is the big? Protein, right? Protein, you can talk about the proteins, right? Proteins, and you can talk about the polysaccharide, okay? Protein, polysaccharides, and you can talk about the lipids, and other than you can talk about the DNA and RNA, okay? So these all will comes under the macromolecules, okay? Can you think which are the things will comes under the micromolecules, student? See here, what is the breakdown product of the protein, student? When I break the protein, what is the protein will give? Or think about what is protein is made up of? Amino acid, right? Yes, micromolecules are amino acid. If you break the polysaccharide, what you will get, student? You will get the glucose, right? Sugars, right? Sugar, right? So that is a monosaccharide. Then coming to the DNA and RNA. When you break DNA and RNA, what you will get? You will get the nucleotides, right? Nucleotides. So these all are the micromolecules. Do you have any questions here, student? What is the difference between micromolecule and macromolecule? You came to know that, right? Micromolecules and the macromolecules. There is a exceptional cases there, okay? That is our lipid, okay? That is the lipid, because I have told you before only, it is 1000 kilodalton, greater than 1000 kilodalton, but our lipid is 800 kilodalton, Okay, our lipid is 800 kilodalton, but then also we are kept in the macromolecules because it is insoluble in acids. So that is the reason we are kept in the macromolecules. Okay, so other than this thing, do you have any questions? If you have questions, you can ask me. Kalyani, Shubhlakshmi. You can ask me, student. Okay, be interactive in the class. Okay. Let us talk about carbohydrate. Student, you all ate what you had lunch? At least tell me this also. Did all of you had the lunch? Did all of you had the lunch? Sandhya, Barsha, answer me. Yeah, I think everyone had, right? Yes. So I am asking you means in the lunch we have ate. Very good, Sandhya. Very good. Yes. Sandhya told yes. I think no one ate other than Sandhya. Barsha is also telling. Okay. Yeah. Student, whenever talking about the carbohydrate, so just we will tell that rice, right? In the rice, the carbohydrate is more, right? In the glucose. So just tell me in the type of things, what are the things containing the carbohydrates? Okay. So sugar containing, not only the sugar containing. We think that carbohydrate means it is sweet in nature, right? No. Some carbohydrates are not sweet in nature. Okay. Yeah. So let us talk about the carbohydrate. Okay. Student, why we will call it as a carbohydrate here? First, we will learn that. Okay. Carbohydrates. Okay. So means it is having carbon, it is having hydrogen, and it is having oxygen. Right. So here, why it is called as a hydrate? Okay. So think here in the water, in the water, what will happen? What is the ratio of the hydrogen to the oxygen, student? 2:1. 2:1, right? In the carbohydrates also, what is the ratio of the what is the ratio of the hydrogen to the oxygen? So that is 2:1. Okay, if you take example, what is the formula of the carbohydrate? CnH2nOn, right? I will write the example of the glucose. Okay, see here, C6H12O6. You take the H12 and the O6. H12O6. This is 2, this is 1, right? So that is 2:1, right? In the water also it is 2:1. And in the carbohydrate also it is 2:1, right? So that is the reason they will call it as a hydrates of carbon. Okay. So because hydrogen and oxygen ratio is same as in water. What is the general formula of the carbohydrate, students? General formula, it is CnH2nOn. It is very important. Okay. The ratio, see here, it is 1 means it will be 1. It is 2 means it is 2. If it is 1 means 1. Okay, like that. So let us talk about the carbohydrate. So it is also called as polyhydroxy aldehyde, and polyhydroxy ketone. Okay, student, in the carbohydrate, we can see the aldehyde also and the ketone also. Aldehyde sugar also, ketone sugar also. For the aldehyde sugar, what the example can you give, student? Glucose, right? You can give the glucose. For the ketone sugar, what you will give? You can give the fructose, right? Okay. So in the carbohydrate, what will happen? So here you can give the sugar's name. Okay, 1 minute. Triose, tetrose, and pentose, and you can take the exose. So here you can take the triose sugar you can tell, and the tetrose sugar and pentose sugar and the hexose sugar. Thiose means it is having the 3 carbon. Tetrose means it is having the 4 carbon. Pentose means it is having the 5 carbon. Hexose means it is having the 6 carbon. Can you give me the example of pentose sugar? Can you give me the example of pentose sugar? For the 3, I can tell that glyceraldehyde. Glyceraldehyde. Okay. And for carbon, I can tell the erythrose. Okay. For the 5 carbon student, can you recall? For the 5 carbon sugar, can you recall? This is a part of the DNA. It is a part of the DNA. Can you remember? It is a part of the DNA. Just I am telling you, it is a part of the DNA. Ribose, right? For the hexose, you can take an example of glucose or fructose. Right, student? Okay. Student, here you have to remember that the difference between glucose and fructose is simple. Glucose is having the aldehyde group. Fructose is having the ketone group. That's it. Other than that, molecular formula is same for glucose and the fructose. So that is C6H12 only. Okay. Yeah. So when we come to the carbohydrate, there are different classifications are there, right? In the carbohydrate. Can you recall, student, what are the classification of the carbohydrate? What we will call carbohydrate as the saccharides, right? We'll call the carbohydrate as a saccharide. Saccharides means nothing but sugar. Okay. Okay. So in the carbohydrate, there are classification, 3 classification. One is the monosaccharide. Okay, monosaccharide. And second one is, can you recall? Oligosaccharide, student. And third one is polysaccharide. Third one is the polysaccharide, right? Okay. So monosaccharide, oligosaccharide, polysaccharide. So why we are calling this as this all the name? Think here. Mono means having single carbon. Okay, that's why we'll call it as the monosaccharide. Saccharide means sugar. The sugar is having single carbon. Okay, sorry, single sugar. Monosaccharide means single sugar, not single carbon. Single sugar. Okay. Oligosaccharide means it is consisting of 2 to 8 sugar molecules. Okay, it is having 2 to 6 sugar molecules, 2 to 8 sugar molecules. And coming to the polysaccharide, it is having more than 8 sugar molecule, Okay. So can you give an example of monosaccharide, student? Which are the things will comes under the monosaccharide? We have talked about the glucose. I have told you example of fructose. In the exam, they may ask you in the multiple choice. They will give you the 4 sugars name. And in this, you can remove the which one is not the monosaccharide. On that time, you have to remember this name. Okay. Fructose. Galactose. Can I write the lactose, students? Girls, can I write lactose? Lactose is a monosaccharide, right? No, very good, Sandhya, very good. No, lactose is a disaccharide, right? So, lactose is the disaccharide. I can't write that, right? Yes, I can write the galactose, glucose, fructose. Then, other than things, can you tell me? Ribose also I can write. Ribose. Then, mannose. Not maltose, okay? Mannose, right? These are all the things I can write in the monosaccharide. Then coming to the oligosaccharide, it will be a disaccharide. In your syllabus, they are given only disaccharide, student. If the 2 sugar molecules are there, means we will call it as a disaccharide. If the 3 sugars are there, means we will call it as a trisaccharide. Very good. Very good, very good. Yes, Sandhya. So these all the things. Which are the things in comes under the disaccharide? Wait, something is happening here. Okay. So in the disaccharide, Sandhya told lactose. Very good, Sandhya. Lactose. And you can take the maltose, right? Then I can tell sucrose. Can I tell sucrose, student? Sucrose, right? With all the three, okay? Lactose, maltose, sucrose will comes under the oligosaccharide. That too in the disaccharide. It is consisting of the two sugars. Okay. So now tell me, what is this lactose is consisting of? Lactose. Lactose is consisting of glucose plus galactose. Sorry. Yes. Glucose plus galactose. Then we can talk about the maltose. Maltose is consisting of, student, glucose plus glucose, right? Then if I talk about the sucrose, or I can tell as the cane sugar, okay? It is called it as the cane sugar. In your sugar cane, you can see the sugar, right? That is the cane sugar. So sucrose, here it is the glucose plus fructose, right? Yes. So this all the example. Yeah, I'm getting the answer. Very good, Sandhya. Yes, glucose and galactose. It is the lactose. Okay. So then coming to the polysaccharide, I have told you that it is made up of the more than 8 sugar, right? So can you recall there are two types? One is homopolysaccharide, and another one is the heteropolysaccharide. Okay, heteropolysaccharide. Let us learn about that in the next slide. Okay, let us move things. So here, student, here I have given the sugars with the open chain structure. Very good, cellulose. Very good, Sandhya. So here we are talking about the 1 minute. Yeah. So this is called as an open chain structure. Okay, student, this is called as an open chain structure. Okay. So see here, glyceraldehyde. So here, glyceraldehyde. So here, see here, you can, it is the aldehyde group, right? It is the aldehyde. Glyceraldehyde is the aldehyde. So here you can see dihydroxyacetone. Dihydroxyacetone is example for the ketose sugar. And the glyceraldehyde here given same thing. And pentose sugar, that is a ribose. So it is having the 5 carbon, right? This is also the aldehyde. See here. And hexose, it is having the carbon. So hexose, how many carbons it is having the hexose? 2, 3, 4, 5, 6. Okay, right. So this is about the open chain structure. In the biology, they will not give the any open chain structures. Okay, but just remember, it will be useful in the chemistry also. Okay, open chain structures. Remember the they can ask you the triose sugar. It is made up of how many carbons? Pentose sugar, how many carbons? These all the things you have to remember. Okay, so see here, this is the closed structure. Okay, so this is, we call it as the closed structure or ring structure. Closed structure or the ring structure. Okay, see here, they are given the glucose. See here, student, glucose. Here you can see in the glucose, here only is the CH2OH group. Okay. So, but in the fructose, you can see here, in the fructose, here is one group. And in the second carbon is also the one CH2OH. Okay. So it is the ketone sugar. Okay. Then coming to the galactose, it looks similar as the glucose, but there is a differ in the which carbon? There is a differ in which carbon, student, for the galactose to the glucose? Can you recall? And see here, they are given the ribose sugar. Okay. One is the deoxyribose. So that is present in the DNA. And here you can see this is the ribose sugar. So what is the difference between the deoxyribose and the ribose sugar, student? See here in the second carbon. In the second carbon, there is no O group, right? No OH, only H is there. No OH in the second carbon. So that's why we're calling as deoxy. Only H is there, no oxygen is there, right? Deoxy where in the second part, second position. See here in the ribose sugar, in the second position, it is having the OH. Like that, you have to remember. Okay. Okay. So then coming to the this disaccharide I have told you. So this is lactose. See here, it is made up of galactose and the glucose. So what is the bond between them, student? That is beta 1, 4. So beta 1, 4. See here. Here it is the 1 carbon. This is the second carbon. This is the third carbon, fourth carbon, fifth carbon. This is the sixth carbon, right? So here you can see 1 carbon, second carbon, third carbon, fourth carbon, right? So that's why the link is between 1 and the 4. So that's why they're calling it as the beta 1, 4 linkage between what? Galactose and the glucose. Okay. So, and see here is the OH group is free, right? OH group is free. So that's why we will call it as the reducing sugar. Okay. So free hydroxyl group is there. Free hydroxyl group. Free hydroxyl group is there. Then we will call it as a reducing sugar. Reducing sugar. Student, we have to remember here, other than sucrose, okay? Other than sucrose, all the disaccharides are reducing sugar, okay? Sucrose is only the non-reducing sugar. Sucrose is not reducing sugar. This is the important, okay? Non-reducing sugar, sucrose. See here, in the sucrose, it is not having the any OH group, right? See here, it is having the CH2 OH group. So there is no free OH group, right? So that's why we'll call it as the non-reducing sugar, okay? This is sucrose made up of glucose and the fructose. See here, the linkage is alpha 1, 2, right? Okay. So then we'll come to the maltose. It is made up of the glucose and glucose. The linkage is alpha 1, 4. So in the cellobiose, it is the beta 1, 4. Okay. Yeah. Cellobiose or the cellulose we can tell. Okay. Cellulose. And isomaltose. Okay. Isomaltose is not important. Just remember sucrose, lactose, maltose, and cellulose. Cellobiose, not cellulose, cellobiose. Okay. Okay. So then coming to the polysaccharide. So coming to the polysaccharide. So student, I have told you that. So more than 8 molecules, more than 8 sugars, right? Here you can see there are 2 types. One is homopolysaccharide. Homopolysaccharide. In the homopolysaccharide, you are having 2 types. Okay. So one we will call it as the storage polysaccharide. Storage polysaccharide. Polysaccharide. And second one is the structural polysaccharide. Structural polysaccharide. Can you recall which is the example for storage polysaccharide, student? Can you recall? Okay. For the storage polysaccharide, in the plants, it is starch, right? Starch. And when it comes to the animals, it is glycogen. It is glycogen. And here you have to see here. So starch will react with the iodine. Okay. Starch will react with the iodine and it will give the purple color. But cellulose will not react with the iodine. Okay. So that thing you have to remember. Okay. Very good, Aliva. Very good, Aliva. Yes. Glycogen in the animal. Yes. So then coming to the structural polysaccharide. So coming to the structural polysaccharide, here also you can see cellulose. Cellulose, student. Cellulose is made up of glucose unit, right? Cellulose is made up of the glucose, okay, glucose unit. So, and coming to the chitin. Chitin is also the homopolysaccharide and it is made up of the N-acetyl glucosamine. N-acetyl glucosamine. It is a homopolysaccharide. The chitin is a structural polysaccharide. I think you have seen on the cockroach and you can see in the this thing, fungi, right? Fungi cell wall is made up of the chitin, right? So here it is made up of the, because of the N-acetyl glycosamine or glucosamine. Okay. So then we'll come to the heteropolysaccharides. So that is the heteropolysaccharide example. Student, have you remember what is the cell wall of the bacteria? What is the cell wall of the bacteria is made up of, student? Can you recall? Can you recall what is the cell wall of the bacteria? Peptidoglycan, right? Peptidoglycan, right? This peptidoglycan, I think you know this, right? Peptidoglycan is made up of NAG and NAM, right? NAG and NAM. That's nothing but N-acetyl glucosamine, and the N-acetyl muramic acid, right? So this is what we call it as a heteropolysaccharide. So student, the simple example is here. So this is one sugar, this is the color blue color, and the another sugar will be yellow color. Okay, so the another next thing will be the blue color, next thing will be the yellow color, right? So here you can see the different types, right? Hetero means it is made up of the two different, okay, or not only the two different, three different also you can take. Okay, yes. So this is about the homopolysaccharide and the heteropolysaccharide. If I want to talk about the starch, okay, student, starch is made up of the two things. One is the amylose, and another one is the amylopectin. Okay, these are two component of the. This is the two component of the starch, amylose and the amylopectin. Okay, glycogen is a single chain. Okay, in the glycogen you can't see any branched structure. Okay, but in the starch you can see the branched structure. In the starch only you can see the branch structure. In the glycogen also it is not there, and in the cellulose also it is not there. Okay, so the branching, it is helpful for the alpha 1,6 linkage. Alpha 1,6 linkage is helpful in the branching. Okay, so student, now you understood this thing. Do you have any questions here in the polysaccharide? Ask me if you have any questions here. If you don't have question, means you can tell me no. No, ma'am, we don't have any questions. Very good, Oliver. Okay, so then we'll go to the next. So let us come to the protein. Okay, very good, Sushri. Thank you for answering me. Let us go to the protein, student. Okay, so student, protein is made up of the amino acids, right? So protein is made up of the amino acid. We'll call it as the building block of protein is amino acid. Building block of protein is amino acids. Okay, yes. So when it comes to the amino acid structure, we'll talk about the amino acid. I told you protein is the macromolecule, right? But amino acid is the micromolecule, right? If you take one bead, 1 minute. If you take like this, right? Like this, if you take like this, so this is, we call it as a protein, not only this thing, okay? Continued structure. This we call it as a protein, but here if I cut this means, this we call it as the amino acid, right? Okay, so let us talk about the first amino acid, okay? See here, student, the amino acid structure is very simple. Carbon is there, and here hydrogen group is there, here R group is there, and carboxylic group is there, and NH3 plus is there. Okay, see here, why we will call it as the amino acids. See here you can see the amino group, right? Amino group, and here you can see the acid group, right? So that's why they will call it as the amino acids. Okay, amino acids. So student, the amino acid we will call it as a Zwitter ion. Why we will call it as a Zwitter ion? See here. So the positive group is there, negative group is there, right? So that's why whenever in the amino acid plus and negative group is there, that we will call it as a Zwitter ion. Okay. So you can take an example here. So here you can see the only H group is there. Sorry, plus group is there, right? Here it is hydrogen group is there. So, but here you can see, here it is having the NH3 plus and COO minus, both the things are there. That's why we'll call it as a Zwitter ion. And here you can see only minus group is there. Okay. It is based on the pH. Based on the pH, the hydrogen will be removed from both the carboxylic group and also the amino group based on pH. Okay, it is based on pH. Okay, whenever there is a decreases in the pH, whenever there is increases in the pH, the H group is going to be removed or added. Okay, so see here, student, here they are given the tree structure. It is very, very important. In the exam, they may ask you the structure and they will ask you which is the structure. Okay, so glycine, see here, glycine we will call it as a achiral. Okay, achiral. Okay, see here, it is having the all the amino acids having the 4 different groups. See here, NH2 group is there and COOH group is there, H is there, CH3 is there, right? But only for the glycine, 2 hydrogen is same is there. Okay, so that's why we will call it as a achiral. When all the 4 groups are different, we will call it as a chiral. Okay, chiral. So that's why we'll call it as a glycine is only the achiral. Okay, and also the simplest amino acid, very simplest, right? See here, only the H group is there. So student, here you have to remember the different amino acids are differ only here, only in the R group. Okay, only in the R group, not in any groups. Okay, these all the things are constant: amino group, H group, and the carboxyl group. See here, when you're talking about the glycine, it is having the hydrogen group, right? When you're talking about the alanine, it is having the CH3 group. Okay, methyl group, CH3 group. In the exam, they may ask you, which is the methyl containing amino acid? Which is that? Alanine. Okay. Then coming to the serine, it is having the CH2OH. CH2OH. You have to remember like that. Okay. One is H containing and another one hydrogen containing, another one is a CH3 and another one is a serine. For the CH3, they have added the OH. CH2OH it become. Okay. So that is how you can remember serine. And other than student, so there are 20 amino acids are there. Okay. 20 amino acids are there. So which is useful in the protein synthesis, Other than the 20 amino acids, so many amino acids are there. Okay. For example, you can take the GABA. GABA will use it as the neurotransmitter. Okay. But it is not useful in the protein production, protein synthesis. Okay. In the protein synthesis, only these 20 amino acids are involved. Okay. Fine. So let us talk about the proteins, which are the amino acids are there. Student, don't confuse with this thing. Okay. Don't worry about this. Okay. So just here you have to remember only the sulfur containing amino acid. Which are the sulfur containing amino acid? They may ask you in the multiple choice. So one is cysteine and another one is the methionine. Okay. But student, have you remember the disulfide bond in the insulin? During the biotechnological application, I have told you disulfide bond, right? That is helpful by only the cysteine. Okay. That is going to be done by only the cysteine disulfide bond, but not by the methionine. Okay. Which are the sulfur containing amino acids? One is cysteine and another one is the methionine. Very, very important. Okay. And then coming to the here, they may ask you this thing also. Basic amino acid. Which are the basic amino acid? You can tell arginine and the lysine. Arginine and the lysine. This thing is important in the when you're talking about the histone protein in the molecular basis of inheritance. Okay. Yes. Arginine and the lysine are the basic amino acid. Other than that, the things are okay, not more important. You can remember the acidic amino acid, aspartic acid, and glutamic acid. See here, in the name only it is giving acid, right? Aspartic acid and the glutamic acid. And simplest amino acid is the glycine. Okay. So like this, you can remember. And the aromatic amino acid is also important. See here, phenylalanine, tyrosine, tryptophan. Okay, this much. If you remember this much, this is more than enough. I will share these notes. You can remember the 20 amino acid. It is not possible to remember the 20 amino acid. Means just remember sulfur containing basic amino acid and the acidic amino acid. That is enough. Okay. So there are two types. One is the essential amino acid and the non-essential amino acid. What do you mean by essential amino acid? Student, if you want to buy anything to the home, we will make a list, right? Are you going to make the list which is already present in the home? No, you are not going to make a list of which is already present in the home, right? So that is non-essential, right? It is already present in the home, right? So you will make a list of which is not there in the home, right? So here also, the body will synthesize the amino acid, right? That amino acid, we will call it as a non-essential. Why we require from the outside? It is already prepared in our body, right? It is synthesized in body, okay? So this thing, essential amino acid, we have to take in the diet, right? Because not present here. So that's why we are taking through the diet, okay? So this is about the essential amino acid. Then coming to the protein conformations, okay? Then coming to the protein conformation, student, remember here, when we are talking about the protein conformation, there are three types. There are four types, okay? One is the primary structure. In the primary structure, just remember, student, amino acid, okay? Only the amino acid. It is made up of the amino acid. Student, between the amino acid, what is the bond is there between the two amino acids? What is the bond is there between the two amino acids? Very good, Sandhya. Yes, sequence of the amino acid. Yes, so it is nothing but the sequence of amino acid. 1 minute. Yes, it is made up of sequence of amino acid. So here the bond is called it as a peptide bond. Okay, so it is a primary structure. In the primary structure, no protein is functional. Okay, for example, if you take an enzyme and those all the things, it must work in the tertiary structure, not in the primary structure. It is only simple structure. Okay, and coming to the secondary structure. So here, student, here you can see two types. One is the helix. One is the helix structure and another one is the beta pleated sheet. Okay. When it comes to the helix structure, you can observe here. So it is just like this. Okay. This is a amino acid sequence. It will helix like this. So this is, we call it as the coiled like this. Coiled-like structure, we will call it as the helix. So normally you can see right-handed helix. Okay, right-handed helix. For example, you can take an example of keratin. Keratin protein, which is present in our skin. Okay, and hairs, you can take the keratin. And when we're talking about the beta pleated sheet, shape of the amino acid. Yes. So see here, in the helix structure, what will happen? Only the coil-like structure. Coil-like structure, we call it as the helix. The example is keratin. So then coming to the beta pleated sheet. See here, it will be pleated like this. Okay, beta pleated sheet. Okay, in between this, there are the hydrogen bond is there. Okay. So here, student, which is the main bond present in the primary structure, means which is the bond? Peptide bond. Which is the main bond present in the secondary structure, means it is the hydrogen bond. Okay, it is the hydrogen bond. In the helix, it is the intra-hydrogen bond. Intra-hydrogen bond. Whereas in the beta pleated sheet, it is inter-hydrogen bond. Inter-hydrogen bond. What is this inter? What is this inter means? Here it is forming the coil-like structure. The bond is between this helix, right? That is why we'll call it the intra-hydrogen bond. Here you can see between this sheet and this sheet there is a hydrogen bond. That is, we call it as a inter-hydrogen bond. Very good. Yes, Sandhya. And coming to the beta pleated sheet, can you tell me the example, student? Fibroin. Right? Fibroin is the example for the beta pleated sheet. You can see in the silk. Okay, in the silk you can see the fibroin structure. Okay. Okay, this is about the secondary structure and this is about the primary structure. Then let us talk about the tertiary structure. So tertiary structure is the very important. It is the 3D structure. It will show the 3D structure. So it will form like this ball-like structure. Okay, it will form the ball-like structure. See a student, whenever the structure is formed, here this end we will call it as the NH3 end. Okay. This end we will call it as the COO minus. So that is the N terminal and the C terminal, right? N terminal and C terminal. Because why we will call it as the N terminal, C terminal? COO minus is there, amino group is there. At the starting, every amino acid is having the NH3 group. At the ending, it is having the COO minus group. Okay. So here there are mainly forces are involved to make this. Okay. So make this globular structure of the protein. So in this tertiary structure, the protein is functional in nature. Functional. Example, you can take the enzymes, right? Enzymes is made up of the protein, right? It is functional in the 3D structure. There are mainly forces involved are covalent and non-covalent forces. Covalent and non-covalent. Can you tell me which are the covalent bonds and which are the non-covalent bonds? Can you think? Covalent bond is the very strongest bond, right? And non-covalent bond. So in the non-covalent bond, you can see the hydrophobic bond, hydrophobic bond, hydrophobic, and you can see the hydrogen bonding, and van der Waals force of attraction, right? Van der Waals force of attraction, van der Waals, right? Van der Waals force of the attraction. So these all the non-covalent bond. Okay. So because of this, all the forces, the protein tertiary structure will be formed. Okay. So student, here you have to remember, whenever the tertiary structure was formed, if we heated, okay, if we heat that protein, the tertiary structure is going to be damaged. Okay. The tertiary structure is going to be damaged, but the primary structure will be like that only. Okay. The tertiary structure is going to be denatured. Denature, okay, at the high temperature, okay. So then let us talk about the quaternary structure. Quaternary structure means what? Student, here you have to remember. So here also one peptide is there, here also one peptide, here also one peptide chain, right? But here you can see in the quaternary structure, more than one peptide is there in the protein. So that is what we will call it as the a quaternary structure. Quaternary structure is made up of more than one peptide. Okay, remember that. So here I am taking the example of hemoglobin. So student, in the tertiary structure, you can take an example of myoglobin. What is the myoglobin function, student? Can anyone tell me what is the function of myoglobin? Storage of oxygen, right? Storage of oxygen. What is the function of hemoglobin? Transport of oxygen, right? See here, hemoglobin is made up of the 2 alpha chain and 2 beta chain. Okay, 2 alpha chain and the 2 beta chain. So totally it is made up of 4 chains. Okay, it is a 4 chain. Okay. So this is the example of the quaternary structure. Both the quaternary structure is going to be damaged and the tertiary structure is also going to be damaged at the high temperature. The only left behind is the primary structure is not going to be damaged by the high temperature. Okay, student? Do you understood this? Do you have any questions here, students? Do you have any questions here? Okay. So if you don't have any question means, I will launch the NPS poll. You can give me your feedback, valuable feedback. So let us continue in the next class. Okay, student? Give me your feedback before going. Okay? Yeah. Take care, students. Read well. Prepare well for the exam. All the best for you all. Thank you, Sushri.